So basically, with cells, of course, you're expected to just kind of know prokaryote versus eukaryote. Like that's not supposed to be something you have to struggle with. You're supposed to know like which organelles are in autotrophs, like plants and some algae that are photosynthetic. You're supposed to just like know that. Like they don't expect that to be a struggle for you. Okay, so of course we're looking at chloroplasts in anything autotroph because they make, they do photosynthesis. And so just knowing that if there's a chloroplast present, that you should kind of already narrow it down to what kind of organism that is. Like you shouldn't have to struggle with that. So of course that's kind of the beginning part. I'm going to put a what you'd call a textbook plant cell as ours, because plant cells have all the organelles, where heterotrophic organisms like animals and fungus and those kind of things don't. Like they don't have chloroplasts and they don't have cell walls and stuff like that. So I'm gonna start off drawing like a little cell right here in the middle of the whole paper. Actually, there's a little spot at the bottom that you might want to. We're gonna write underneath it just a little bit of stuff. So basically, we're gonna have a little blown up area here of the cell membrane. So you need some space here, and you need some space here, and some little space here. But that's about it. Okay. So give yourself some margin room, and maybe draw your cell like up and down like this instead of sideways. Okay, so first thing I'm going to do is make sure that we have a cell wall here, because this is supposed to be a plant cell. So kind of thicken up our cell wall. We didn't talk much about cell walls. They actually kind of are just a structural thing. They, of course, have to have some sort of like openings in them, like pores, so that stuff can get through and proteins can reach through the cell wall into the next cell or whatever. But they're made of well, actually, let me just finish drawing the little thing here. And so then right inside of there, I'm going to draw a wee little line there that's going to represent our cell membrane. Okay. And I'm going to enlarge the cell membrane so we can, it's pretty important as far as AP Bio goes. So I'm going to have a little inner membrane here, a little cell plasma membrane, they call it. Okay. All right. So then I'm going to stuff the nucleus in the corner, because that's the way plants are. I'm going to draw a nucleus, and then I'm going to put some DNA in it. Of course, DNA isn't actually that big in the nucleus. That's pretty big DNA. And then I'm going to be like, draw the DNA. And then you need to know, and if you need to write it because you forget it, you need to know that DNA never leaves the nucleus. Okay, it's stuck in the nucleus. So if anything is happening with DNA, besides mitosis, because in mitosis the nuclear membrane breaks down, but besides mitosis, if anything is happening that has DNA involved, then that's happening in the nucleus. Okay, so you can just kind of put that in your brain. If you see DNA, if transcription is occurring and the DNA is making an mRNA, well, that's in the nucleus, because the DNA can't leave the nucleus. Okay, so just kind of compartmentalize your nucleus into that. Okay, so as we kind of get through this, I'm going to number some organelles and draw them in here. So as we go, I'll be adding organelles to this. Actually, funny story, nucleus isn't even on your list of organelles to know. Like it's not even in your standards. So I'm not going to write any details about the nucleus, because apparently we don't need to know any for the test. But I mean, that's strange, because the nucleus does show up on in pictures and stuff. Yeah.
The prokaryotes don't have a nucleus, right?
Prokaryotes do not have a nucleus. That is correct. Okay, so let's see. Let's start with that. Let's start over here on the side, and we're gonna go, prokaryotes versus eukaryotes as far as this stuff goes, because we need to make sure, if I can spell today, that's a Y there, Y-O-T-S. Okay, so prokaryotes versus eukaryotes. So the first thing we kind of have to know is what's the difference, right? What's the difference? So we've already dealt with the difference in DNA, right? Circular, single, no heterozygous when you come to mitochondrial traits and things, because mitochondria DNA is just like prokaryotes, right? So that's kind of what we dealt with yesterday as far as nucleic acids in prokaryotes versus eukaryotes. So what about organelles? Okay, these actually, okay, I'm going to put organelles here because eukaryotes have them. And if you want to make like a little T chart here, maybe I should separate them. And then prokaryotes have regions. So even though they don't actually have membrane-bound organelles, like a membrane around the organelle, they do have areas. Like they have a nucleoid region, they call it. That's where the DNA is. It's kind of in the center. They have, of course, the cytosol or cytoplasm, whatever you want to call it. But that's, once again, not in your standards either, so we don't have to put that on there. But they have, remember, I'm going to write it again, though I don't know. I think you should know it by now. So circular, single DNA. If you've already, or you're like, I'm good in this, well, then don't write it. Linear, paired DNA for that guy, okay? And then remember that with endosymbiosis equals organelles. Now I'm gonna start listing organelles. So that's where I'm going with this. Endosymbiosis equals organelles. Okay, so one thing they do have in common, if you have a little spot right here, is they both have ribosomes. Oh, I need to make my box bigger. Okay, so they both have ribosomes. Can you see that? It's probably tiny. Sorry, that's so tiny. That's what I said. I need to make it bigger so you guys can see it. It's annoying. That says both have ribosomes right underneath it. Okay. And so as we came up with some endosymbiosis back in the way back in the primordial soup days, yes.
Do prokaryotes have that intron, extron, exon thing?
No. And that is what we'll get into that. I don't know what unit it is for College Board. It was our unit 2, but I don't know what it is for unit maybe 4. But we'll get into that in more depth then. So why are organelles evolutionarily beneficial? Like that's kind of what we need to know, because that was a question on one of our, it was whenever there was a chloroplast on unit 5. You had a chloroplast and you were supposed to talk about like what to deal with these membranes and the thylakoids and all that jazz. Like what's the benefit? And remember, the only reason that you would see things be kept like this, like, still have organelles, and all eukaryotes basically have organelles and pretty much the same ones. And so there must be some benefit, or else we wouldn't have kept that. And so what's the benefit? So that's kind of what we need to have on here. I actually have it written up here, but I don't know if I have enough space. Do you guys have any space about yourself? No? Okay. I need to put maybe over here somewhere, or just right underneath here. I'm going to write the benefit. There's three benefits that cells have from having organelles. And so I'm just going to list 3 things. Okay. So find a spot to list 3 things. Maybe I'll put mine right here. So benefit, and you can do it underneath where it says endosymbiosis is organelles. That's fine too. Benefit of organelles. Now, not all cells have these membrane-bound organelles, so clearly they're not like mandatory. Like you don't have to have organelles in order to be successful, but there has to be some benefit, or else they wouldn't continue. So one benefit is that you compartmentalize the cell, and that's kind of the number one that we always hear about. Okay, let's talk about compartment. That's a really long word. Compartmentalized. So compartments in a cell are important. Well, let's just consider this. The prokaryote's about the size of mitochondria, right? So compared to a eukaryotic cell, prokaryotic cell is really, really small, right? And so you don't have to like go far to find what you're looking for in a prokaryote, right? So if you're trying to do some cell respiration on the inner membrane, and you're, because that's where prokaryotes do them, in that folded inner membrane. If you're trying to do some cell respiration, the glucose isn't far away because it's the size of mitochondria, tiny, right? Not so with our lovely eukaryotic cells. So compartmentalization allows for basically the things that you need for whatever process it is, because it's very specialized. You got your mitochondria, your lysosomes, your ER, they're very specialized with what they do. And this allows you to keep the things that you need to do your processes close at hand. They don't just go, diffusing through the cell because, high concentration, low concentration, there they go. So they don't just go diffusing across the cell and you're like, wait a minute, I can't, you're far away now. So that's kind of the thing. So compartmentalizing actually allows the cell to kind of do its work, right? It's like, otherwise it might be really far away and it would be slow. So compartmentalizing allows for efficiency, to have materials at hand, if you will, okay? This also reduces competition, which sounds kind of weird, within the cell. But same kind of idea, like don't use my stuff, right? So if you have stuff you need, these could be oxygen, carbon dioxide, lots of different things, ATP, lots of things that you need. If it's compartmentalized, it can be right there at hand rather than being like, okay, well, great. Now the mitochondria just literally used all of our oxygen. Thank you so much, mitochondria. We needed some over here too, but you do you. But actually compartmentalization keeps it where those things are separated. Like they're separated so that they're not like fighting for resources and the resources can be delivered to the correct place and they just stay there and yay. Okay. So we don't have to like have a competition between our processes in the cell. The cells are extremely busy, right? Every part of the cell is working all the time. It would decrease your energy efficiency if you were constantly using other organelles' stuffs. But if you can keep it compartmentalized, then everybody kind of has their own stuff and it works a lot better. Does that make sense? Okay. And then the last one here, our third reason that you would have a benefit is to just increase surface area for some things, right? So not all things require an increase of surface area, but we talked about this with mitochondria, right? It has that really highly folded membrane, and that's increasing the surface area. If it didn't have a membrane-bound organelle, couldn't do that. Inside the chloroplast are those little stacks of thylakoids, and they're just these little rings increasing surface area, right? So that helps the organelle to do its work. So those are three solid reasons that eukaryotes benefit from having our organelles. So anyway, and that's come up several times in our practice exams. So perhaps that's something that's more important this year than it has been before. Okay, so let's start walking through our organelles here. Which ones do what kind of thing? I have an order to it, so I better keep my order. I'm going to forget somebody. So first of all was our ribosomes. Okay, so I'm going to just draw a few ribosomes. We're going to have a big vacuole kind of in the center. So just kind of stay out of the center a little bit. Just put in a couple ribosomes around the edge. And then I'm going to also put in our endoplasmic reticulum. So that's kind of a weird little hallway looking thing. Okay, and so it goes right around the nucleus. But I'm putting that in because there are some ribosomes on that as well. There's actually two parts of the endoplasmic reticulum, and I can't really figure out how to differentiate my drawings of them. So I'm just going to put some ribosomes on one and not the other, and we'll call it solid. They're kind of all meshed in there together. Okay, so this is our number one organelle right here. I'm going to put a number one beside it. There he is. Well, I just totally zooped in my ribosome in that circle. And so I'm going to put him right here. Okay, I'm going to put okay. First thing, ribosomes. And we're going to have about 5 organelles that you have to know. Ribosomes, first of all, they're not membrane bound. So kind of straight out the gate, we have one that's atypical, but that's okay. They are simply rRNA. rRNA mixed with some proteins. But, hey, proteins are everywhere. Okay, that's all they are. And they are in charge of protein synthesis. And you know this. Like, I don't have to tell you that. They create proteins. Okay, so that's our number one organelle. Of course, we notice right here, if you want to put a little 1 beside this note here, over here, I don't know if you forget that they both prokaryotes and eukaryotes have ribosomes. So I was kind of like, oh, dang it, I probably shouldn't have put it underneath the organelles because that kind of seems like it's only for eukaryotes when it's actually in both. All organisms have ribosomes, highly conserved, extremely highly conserved. So that means that it must work well. Like literally all organisms, like every single one has ribosomes. If you think about that, that's pretty crazy. Because that's like that and DNA are like the only things that we all have in common, bacteria and everything. Okay. But our prokaryotes have a slightly different type of ribosome, like their RNA is different, just like their DNA is a little different. So that's cool. All right, cool. Number 2. Any questions about ribosomes? No? Okay. Number 2, we're going to go ahead with our endoplasmic reticulum. I'm going to call it ER. You can write it out if you want. I don't want to write it out. We have 2 little ones here, so I'm going to say rough on the left, and I'm going to say smooth on the right. Don't need to know too much about either one, but just that that they actually have 2 different jobs. They want you to have an overview of the smooth, but not really know much about it. Either one really. So the rough endoplasmic reticulum helps with transporting things. It's like a little hallway. And then of course, I hate writing this on here. Protein synthesis, but it's because of the ribosomes, right? So I'm going to put that in little bitty thing there, because the endoplasmic reticulum isn't actually doing the protein synthesis work. It says protein synt, S-Y-N-T-H, and then it says because ribosomes, B/C, because ribosomes, because they're not, the proteins are actually being created by the ribosomes that are bound to the endoplasmic reticulum. The endoplasmic reticulum is just kind of facilitating the transport and the housing of them. Yes.
Where does the smooth one go on the drawing?
I just made it out here. That's what I was like. They're actually kind of attached to each other right here. They're kind of all a big mess. But I was like, well, I'm just not going to put ribosomes on one of them. Here, I'll just make it fatter. Smooth one looks a little different, but they're just all a mess around the nucleus. So this is our number 2 right here. So rough has the ribosomes and smooth doesn't. Smooth is actually, it creates lipids and detox. So it's in charge of creating lipids and detoxing.
And it also is in charge of transporting.
Yeah, it does some transport. Yeah. Transport of lipids. But that's another thing, is a lot of your standards is like, oh, but beyond that, you don't have to know anything about the smoothie ER. I'm like, okay, just know that it has some lipid. Know that, I think what you should know about the smoothie ER is it does not have any protein synthesis function. Okay, there's no ribosomes on it, and the ribosomes are what's actually making the proteins. So no proteins for the smoothie ER. It's all about lipids and detox. Okay. All right. Number 3, we're going to go into our number 3 and 4. We're going to go into our, I'm going to go ahead and draw my little vacuole on here so that I can know where I need to be. I'm just going to make it kind of a little sort of circular rectangle. We're going to deal with that in a second. But I need to know where my space is. So I'm going to draw my little mitochondria that look like little jelly beans. I should put a couple on there. And then I'm going to draw my little chloroplasts that look like, I don't know what, little turtle shells. And if you have colors, you can make your chloroplast green. I mean, I guess I could find a color, but I don't know what to do with them. Okay, I'll make it green so that you can differentiate. Here's my little guys. Okay, remember, these are autotrophs, so they have both. If we were in a heterotroph, we wouldn't have any chloroplasts. Okay, so number 3 and 4, I'm going to put side by side right here. 3 and 4, because they are 2 energy ones. So let's do mitochondria first. I don't know why. That's just how I have it on my list. Mitochondria and chloroplast. Okay. So mitochondria and chloroplast have something in common. They both have a double membrane, and that is mentioned over and over again. So it's mentioned in endosymbiosis. It's mentioned in the structure of the organelle. It's mentioned in when you're talking about what they do as far as photosynthesis. So it's mentioned in like 3 or 4 different units. So make sure you know that mitochondria and chloroplast have a double membrane. Like they have the one that they call like a vesicular membrane. So a vesicle, you know, like a vesicle is like when something is like carried, it's got a membrane around it, like the cell is like, hey, this doesn't actually belong to us, so let's just put it in this little vesicle. So because the vesicle is ours, so now we know it's supposed to be here, a little transport vesicle. And so on the outside of their double membrane, that outside one, you'll often hear it called a vesicular membrane because it looks like a vesicle. It's like the mitochondria and the chloroplast have been wrapped in a vesicle, which is why they're like, endosymbiosis. And it's actually wrapped in a vesicle at one point. So that's kind of the double membrane thing. Here you need to know about cellular respiration. I'm just going to abbreviate that. And then, of course, over here is photosynthesis. Okay. I can't draw in the different little parts of the mitochondria and chloroplast. They're not big enough for me to draw them in. We will get into that a little bit more whenever we do energetics. But if you wanted to add it here, you could add in, the matrix for the mitochondria, the inner membrane, the thylakoids, and the stroma. And you could put kind of what occurs in each. We will revisit that in energetics. Okay, so if you're like, I'm cool with just adding that in energetics, then that's fine. Okay, so those are our energy organelles. Okay, and then we need to get our little lysosomes in here, and I think then that will do it. Let me double check. Ribosomes. Okay. Yep. Okay, so number 5 is lysosomes. If you have space for it, still down here. If you don't, you can put it where you need to. Oh wait, I need to put in the numbers on these guys. So chloroplast is 4, and mitochondria is 3. Okay. And then lysosomes are just going to be like, they're just going to look like that. So we're just going to go here. I'm going to color them in black. As soon as you hear lysosomes, you should think lysis. Lysosomes, lysis, breaking stuff. Vesicles, they're not on your list. The vesicles are whenever you are bringing in stuff from the cell or bringing stuff from the Golgi. Oh, Golgi. Did I totally not have Golgi on here? We got to put Golgi on. We'll have a number 6. But the vesicles are the little like trucks. Lysosomes, they are for lysing things. They're full of lytic enzymes. Sorry, this is getting really low. Probably can't see that. See, I always have to figure out how to do this whenever I'm with you guys, and then I get better at it as the day goes on, so I apologize. Okay, so lysosomes, that's a lysosome right there. You should just think lysosomes, lytic enzymes. Well, gosh, that's enzymes that break stuff up, because lyse and lytic and all that, that's all about breaking stuff, right? We talked about that yesterday. If you hear lysing, hydrolysis, that's what we did yesterday. Hydrolysis, anything that has that lyse in it, it means to break it. And so lysosomes are all about breaking stuff up. And so they are going to digest stuff. That's what a lysosome can do. It can digest with its lytics. It can apoptosis, right? Can kill the whole entire cell. And it can recycle stuff. So if say a protein is broken down, it can break all the bonds and then reuse the amino acids necessary. So it's all about destroy, recycle, destroy the whole cell, apoptosis. So that's kind of our lysosomes there, okay? All right, I'll stick Golgi right underneath here. Golgi apparatus, Golgi body, Golgi, whatever, just no Golgi. And if you actually just write Golgi, they're okay with that. Like, you don't have to write out the whole thing, but whatever you want to have a word. They either call it Golgi apparatus or Golgi body is its full thing. Golgi is just awesome, honestly. So let's put a Golgi in here. It's usually somewhere around our nucleus, though it can be a little further out. Oh gosh, I put a mitochondria right in the way. I'm moving that mitochondria. So find a spot. You can put it up here, around there, whatever. But what it is it looks like a bunch of little stack of little sacs. Weird looking thing. And it has like these little vesicles, and that's what reminded me, because Natalie reminded me of vesicles. So basically this is number 6 here. Basically what you have is, now with the Golgi being a part of stuff, is you have a connected intermembrane system. So basically the Golgi's job is to modify, can't spell, and like ship. In the actual standards it says protein trafficking, and I don't like the word trafficking right now. So modify and ship, I'm going to say, proteins which could mean modifying, could mean folding, if there's any changes that need to be made, things like that. Typically, it's just kind of folding them up and shipping them off in little vesicles. So with the Golgi as part of our routine now, we have a whole intermembrane system, right? So we now have where it can pass from the ER, could be coming out of the rough ER in these little trucks, so these little vesicles here, going and like becoming a part of the Golgi, being passed from little membrane sac to membrane sac of the Golgi, and then passing out of the cell membrane over there. And so now with the Golgi a part of it, it kind of makes it where the whole thing can now be, everything can be moved around the cell, because the Golgi is really pretty good at that. Yeah.
Doesn't the rough ER transport proteins too?
The rough ER will send them off to the Golgi. They have to go, basically the Golgi is kind of your central shipping area. Okay. All right, are we good on that? There's your organelles. Those are the ones you have to know, or you just have to be familiar with them. Like, you don't have to like kill yourself trying to like memorize every little bit of them. Just be familiar with them, like which is going to do what, and kind of be able to just be comfortable if they talk about them. Yeah.
What is the organelle or molecule that gets the protein, the form of protein or whatever product from one place to the Golgi?
Like if we made it on here,
Yeah.
and then it's got to go to the Golgi, these little vesicles, but actually they're just a part of the endoplasmic reticulum. It's like the end of the endoplasmic reticulum would pinch off, and then go to the Golgi, which could then be absorbed into the Golgi. So that's why they kind of call it the endomembrane system, because it's like the membranes, they all share the same membrane. And so you can just be like, stick it on there, pull it off of there. It's like having little, I don't know, dough balls. You have like this, think of like the endoplasmic reticulum, the Golgi, and the cell membrane all as parts of a dough ball that you've made. And it's like you can pinch a piece off there and then smash it into that one, and then you can't tell where it came from in the end. Yeah, Natalie.
Okay, so they make the RNA in like the nucleus, and then they pass it through the endoplasmic, the rough ER. Then it goes to the Golgi, and then it goes to the vesicles.
Okay, careful with it. Are you talking about?
Protein.
Okay, yeah. Careful with those pronouns. Or the whatever you call that. I don't know. I'm not an English person. But basically, the mRNA, yes, is made in the nucleus, right? Goes to a ribosome that's connected on the endoplasmic reticulum. The protein is fed directly into the ER from that. And then as it goes through, it begins its folding, gets in a little vesicle, it pinches off at the end of the endoplasmic reticulum, goes to the Golgi, gets absorbed into the Golgi. Golgi continues to fold it, modify it, tag it for where it needs to go, little vesicle leaves. And that would be for if it's going to be a part of the membrane or if it's leaving.
What's the wording for if it's used in membrane and then leaving membrane? So I remember we had like a FRQ on it, and you made it very clear, like this one goes.
Is there a special word for it? Let's see. I don't know. I'm trying to think. If I were going to say it right now, and there may have been a word that I'm just not in my brain right now. If I was to say it right now, I'd say like a protein that's being embedded in the membrane or is being like exported from the cell. And those are made in that whole system. Now, these guys right here, these little free ribosomes right here, these are ones that where it would go, the mRNA would go there to make a protein that's just staying here. That's staying inside the cell. And so that's typical, unless it's something really, really sketchy, like the lytic enzymes that are in here. And they need to be made in the ER and put in the vesicle because they're kind of dangerous. Like their pH is really low. And so if you carry that through the cell, it kind of has a tendency to not do great things to the cell. So they do. But other than that, and you can just ignore that in your AP writing, just pretend like all proteins that are made for inside the cell are made on these free ribosomes, and all proteins that are made to be embedded in the membrane or out are made in the endoplasmic reticulum. Okay, and that you can just generalize it like that, and that'll be fine. Okay, you don't know the little nitty gritties. Okay, questions on those organelles? We actually have one more that I need to write. I'm just writing it in here because I have all this space. So this is a vacuole, and the vacuole is large in a plant. Well, the large central vacuole is large. There are other little vacuoles that just kind of are around that are kind of just storage. So just think vacuole is just storing things, right? So vacuole and vesicle, all right. Vacuole and vesicle are actually really similar, only a vacuole is going to be like a permanent thing, like in the cell, right? There it is. This little vacuole. It's going to hang out there. It's going to store whatever the heck it stores. Most of our vacuoles are specialized and they have names, but we don't need to know those names. Okay, so depending on what they store, they would have a little name that goes with what they store. But they're a permanent structure. Like a lysosome is actually a type of a vacuole, right? And it's not going to go and become a part of the endoplasmic reticulum or become a part of the cell membrane, right? It is a permanent structure. Vesicles are like temporary. Like it's like a little thing that goes and attaches to other things and gets rid of its stuff. Does that kind of differentiate them for you? So a vacuole is going to stay and do its job for the life of the cell. Where a vesicle is like, oh, I came from the endoplasmic reticulum. Now I'm going to be, like engulfed in the Golgi, and now I'm going to go be engulfed in the cell membrane, and I'm not going to be there anymore. I'm just a part of the membrane now. So that's the difference kind of between vacuole and vesicle. In a plant, there is a large central vacuole, and it's just one of the types of vacuoles, but they do want you to know this one. And basically, it's full of water. So you've got water filled, and this allows you to create, not you because you're not a plant, this allows the plant to create some turgor pressure, which I like to draw like this. And it can be changed. So this can change. Remember what that is?
Water potential.
You can change your water potential, right? Right? Do you remember that part of the water potential formula is pressure potential? Right? So it's, solute potential plus pressure potential. So if you change your pressure inside your vacuole, then you can change your water potential of the cell. Gives the plant a little bit of control beyond just solute concentration. Because plant doesn't have a whole ton of control over its solute concentration. But it has quite a bit of control on how much water it allows into the cell. And it almost makes it feel to me like, oh, the plant has a brain. And it's like, let me have some. Okay, it's all chemistry, but it's interesting. But yes, the plant, definitely they will change how much water is in here depending on if they need water to move it. So it's basically from signals from other cells, like how much water is available, and they'll actually alter the amount of water in the central vacuole. Now, on top of that, it is used for photosynthesis if there's no water available, like if water is not just coming in from a nearby cell. So, times of drought or times when I forget to water my plants or whatever, it can use that water for photosynthesis. So it's like storage of water, and then also can be used to change its water potential to get water to get in or get water to get out if necessary. Okay. So that's a vacuole. Just know that that's not the only vacuole. There are some other vacuoles around, but and they just store whatever they need to store specific to that. Okay, where are we? Got that done. Okay, let's look at our cell membrane and then surface area volume ratio. And that's something I do need to kind of hit hard. Okay, let's find a spot that we can circle on this tiny membrane out here. There's a tiny membrane, and then we're going to zoom it in. Woo. Okay, and we don't need a whole ton of space, but we need a little space. We're going to draw a little membrane, right? Just some little phosphate heads and some little lipid tails. We're going to draw it the little not very exciting way. Make sure, oh gosh, that one doesn't have a head. Make sure you know that, this is the hydrophilic. And this is hydrophobic. Okay, and then we're going to draw some things in it. So I'm just going to draw some little stuffs here. A blob in the middle of the lipids is going to be our cholesterol. If I can spell it today. It's touch and go on spelling cholesterol for me. I don't know if that's spelled right or not. Whatever. So that's our cholesterol. They don't really tell you too much about what cholesterol does. It just kind of stabilizes the membrane. In temperature extremes, the cholesterol is super helpful, but it's just kind of there for that. We have protein, so I'm going to draw a big fat protein that goes all the way through and kind of has some stringy stuff. So there's protein. And that protein is amphipathic. That is so hard to say. Amphipathic. What does amphipathic mean? Do you remember? Remember what amphibian means? Ampha. Both, right? Amphibian is actually an amphibious vehicle. You know this, like water and land. So amphipathic means it has both water parts and the lipid parts, right? So it's going through the water and not. Amphipathic proteins are the ones that fold differently inside the membrane than they do on the outside because of the presence of water. So water on the outside, because you have your cytosol and your extracellular fluid. No water on the middle, so the protein just folds differently in the center. Like the hydrophilic parts will go in, the hydrophobic parts will go out in the middle, which is not how we typically think of a protein folding. Okay, there's also some glycoproteins. What would a glycoprotein say to you? What is glyco? Yeah, it's like a sugar protein combo. So which of our macromolecules was the sugar? Carbohydrate. So your glycoprotein is a carbohydrate protein combo. So you might see those on there. They kind of have a tendency to stick out into the extracellular fluid. What about a glycolipid? Sugar fat. And so you got your carbohydrate fat. They'll be in there too. They kind of do the same thing. They're all about signaling. Those two are. So they might mention glycoproteins, glycolipids. They're all about talking to nearby cells. They stick out into the extracellular fluid and do a little communicating. So if you have space and want to draw them on there, just kind of draw them sticking out the top. I draw it like a little string of pearls, but however you want to draw it. And they're about communication. Okay. Let's see. And you know the phosphate head, right? You know the lipid tail. That shouldn't be a problem. So then down here, I want to know what gets through. So passes freely is going to be small, non-charged stuff like into nitrogen, oxygen, carbon dioxide can all just get through. They don't have to have a channel, right? They can just zip through the membrane, no problem whatsoever. Okay, what about water? Yeah, no, not really. I mean, it can kind of wiggle through at some point, like little bitties, but it would not be efficient. So what does it do instead? What's its protein channel called? Aquaporin, okay? So water is going to use an aquaporin. Okay, though you have to know, or you should know, that some water can slip through, like not a big deal. Like they can sort of wiggle through, but aquaporin. Remember whenever we looked at our membrane we were doing our model and it said like 3 billion or something, or 6 billion water molecules per second, or some ridiculous number of water can go through an aquaporin. But of course that would be not great for the cell. So, okay. So let's talk about some transport here. So underneath the cell membrane, so all this side over here is kind of cell membrane side, because we're going to talk about some transport. How do we get stuff in and out, right? So we have active and we have passive transport, okay? So this one, active is energy required and passive is no energy, okay? Remember that passive absolutely can have a channel, right? Just because it's passive transport doesn't mean it's just moving through the membrane. Passive transport, osmosis is passive transport, but it has to have an aquaporin to do it. So that's what we call like facilitated diffusion, right? Or something like that. But you just kind of need to know the active and passive thing, okay? Let's see. So then we have to know that this is going from active, could go from low concentration to high. And this one, of course, has to be high to low. Active doesn't have to be low to high, right? But if it is going low to high, it is active, right? Does that make sense? You can go from high to low in active. It could just be you have some massive thing that has to get in, and so you can't get it through like a channel. So in that case, we would call that endo. I'm going to just do endo and then exo like that, and then I'm going to hyphenate it and say cytosis. So with endo and exocytosis, you absolutely could be going from high to low concentration. It's just you can't fit it through. And so then you have to have energy to morph that membrane. So remember, endocytosis is when you're pulling something into a vesicle. Yeah? You remember this? Like it hits that thing and it goes and pulls it in. So you have to have energy to do that. Like changing the membrane that much requires energy. And so even though you could have tons of whatever it is outside and none on the inside, it's still going to be active because you're having to morph your membrane. Okay? So it could so this is not a requirement for active. It's just if you see that must be active. This is a requirement for passive. It has to be like that. Okay? Questions on that? Okay. And then as for water, let's look at, I'm going to stick on here. I'm going to put osmosis on here, right? As a form. And so we are going to go from high water potential to low, right? You can write this about 5 different ways. You can write hypotonic to hypertonic, right? You can write low solute concentration to high solute concentration. But whatever you need to know, like this is under the heading of osmosis right here, is you need to be able to write it out. You need to find your way. What are you going to say when they ask you why the cell is increasing in size? And it's because of water. Like you have to have a way. You can either say, well, the water's going from a high water potential area to a lower. Remember, it goes towards the sink, it like sinks in. So high, or you can say it's going from hypotonic to hypertonic. Or you can say it's going from a low solute concentration to a high. But pick your way to say it and say it that way, okay? And you can have all 3 or 4 memorized if you want, but you don't have to. You just have to say it in one way, but you need to say it in a biology way. You either need to use a hypertonic vocab, a water potential vocab, something, but pick your way to say it and know how to say it, because that's a free response question a lot of times. A lot of times there'll be something about water in the free response and you've got to talk about it. And so make sure you talk about it in such a way that they know that you understand biology, okay? At some point, we will go over all the formula sheet formulas. It might be like after we finish reviewing all the content. I might have it one of those days that we have while y'all, whenever we have the least conflicts, we might just have like a math day where we just sit there and go through all the different formulas and work them. I'm not wanting to just work water potential formulas right now, okay? It's just kind of gets a little into it, but we will. Okay, so the only thing I have left to do, I think, is surface area to volume ratio. Let me just double check my notes here. Got ribosomes, chloroplast, mitochondria, Golgi, ER. Okay, so surface area to volume ratio. This is one that I usually do a lab for. And it was one of the ones that the lab just didn't work with our time. If you don't know SA to V, like what that means, then write out surface area to volume. This is something that we hit pretty heavy in freshman year, so I wasn't too concerned about it, but there's just a couple of little tips that I can give you that will help you get these problems correct. This is kind of a math problem that we're not going to work it. So what we've got going on here with the cell and the surface area to volume ratio is you got to have surface area, because that's like your way stuff gets in and out, right? So because the cell membrane is the way you get nutrients in and the way you get toxins out or waste products out, you got to have that surface area. If your volume is, there's a magic sweet spot. There's a sweet spot for eukaryotes that says, okay, we have plenty of volume, because you need volume for all this. Like you got to have a spot for your organelles. I mean, obviously you can't just be, we're going to be the size of mitochondria and pack all this in. No, you're not. Okay, that's why prokaryotes are smaller than eukaryotes, because eukaryotes got all this stuff in them, and it's just physically impossible for them to be tiny, right? So they get bigger and bigger and bigger as evolutionary time kind of goes on. They get bigger and bigger, and then all of a sudden it's like, whoa, too big. Now I can't get stuff in fast enough to fill up all this, right? And so that's kind of what we look at. So surface area to volume ratio, actually the larger your surface area is in comparison to your volume, better off your cell is. But of course we need some of that volume. So it's kind of got to get this sweet spot, right? So cells kind of have a limit to how big they are. We just don't have a ton. One exception, the egg. right? The egg is the biggest cell and it's massive compared to all the other cells. But that's because it has a lot of volume of nutrients and things in it. Like that's the way meiosis has made it. It's got a ton of nutrients in it that it stole from other cells. And so until it divides and divides and divides and divides and divides, it can survive. Okay, it's not pulling in its nutrients from the outside. Special. Okay, all the rest of the cells are like max size. After that, we die. Okay, so surface area to volume ratio, you want a large surface area to volume ratio. You want the surface area to be large. So how do we do these problems? Because they can give you several different ones and be like what's going on? You know, which one is the better size? Okay, so what you do is you just take your surface area. I'm just going to make a little formula here. Take your surface area, divide it by your volume. This is how you do the problem. Okay, and that'll give you some number, right? It'll give you a number basically to 1. Okay, you're like, I can't see that, Miss Woolley. Okay, here's what I'm writing. So for surface area volume ratio, this is what it says right there on the board, down there. So you're basically taking your surface area, divide it by your volume, and that'll give you some number. That's the number you compare to the other ones. So if you have 4 different cells and they give you surface area for each of them, and sometimes they're asking you to calculate it. And on your formula sheet is how to do it. Like every single, like surface area cube, surface area, you know, it's on there. So you don't have to memorize your surface area volume formulas. Super simple to do. You just plug in the numbers, and then you take your surface area and you divide it by the volume, and then they give you a number, and then you compare that to whatever they're asking you to compare it to. The larger one, so the larger this number, for nutrient, like for the cell, so that it can get nutrients in and it can get toxins out, because that's telling you compared to the volume, the surface area is large. Okay, so there was a practice. I don't remember where that was. One of our practice tests that we haven't done it, did this, and it was really a pretty good one. It said that because of some genetic problem, the red blood cells were round instead of that concave thing they do. And then it told you the surface area volume for the concave one, and it asked you to calculate the one for the sphere, for the round one. And then of course you just had to go and do that. And then it said, like, what's the deal here? And you had to say, well, the round one is like has a lot more volume compared to its surface area. So its surface area to volume ratio was smaller, so that was bad. Okay, and so that's basically what it wanted you to do. Okay, so not kind of a big deal there. Surface area to volume ratio is super simple. You just have to remember that once you divide them, you want the large number, because that's the big surface area. So that's the one that's going to allow you to get nutrients into the cell faster and better. Okay, what questions do you have? That's cells, unit 2. The only thing I think we didn't actually do in class was that surface area to volume. But everything else we did at some point. All right, now we got through reviewing units 1 and 2, my 2 least favorite units. The rest of it is just fun. Yeah.